All right, welcome back. Are you going straight through? Did you click immediately over? I really hope you took some kind of a break. Stand up, stretch, call someone that you love and say hi, and then come back. Do a headstand, get some blood to your head. Okay, I'll stop telling you guys what to do. Let's jump back into it. Okay, so we've been moving along, right? We are in irreversible cell injury. The first two steps are the same between reversible and irreversible cell injury. We've just gone through this negative effect of free radicals. We are now on the step of mitochondrial damage. All right, quick pop quiz. What would you say the mitochondria does? Can you say it out loud right now? I mean, in your head or to the person next to you? What's the main thing that pops in your head when you hear mitochondria? I hope you all said make ATP, right? Mitochondria is our ATP making machine, the site of cellular respiration. It takes oxygen, it takes the byproducts of our macronutrients, and it pumps out ATP. During cellular injury, we start to damage our mitochondria. This is a big problem because this entire story started with ATP depletion. Now, any possible ATP that might have been being made by the mitochondria is going to get worse because it's going to get damaged. Okay, first thing that gets damaged is to the mitochondria for the same reason we've actually already talked about. The membrane has a phospholipid layer. The phospholipids are attacked by phospholipase directly, right? That's the targeted attack. They are damaged by this accumulation of reactive oxygen species, what we refer to as oxidative stress. As the membrane gets damaged, as the mitochondria gets damaged, you are going to decrease oxidative phosphorylation. Just in case you're not as familiar with these terms, oxidative phosphorylation is just another fancy way of saying cellular respiration, which is just another fancy way of saying that it's going to be worse at using oxygen to make ATP. Oxidative phosphorylation is using oxygen to make ATP. So decreased oxidative phosphorylation is going to further deplete ATP. Remember, we're talking about these vicious cycles. An initial stimulus causes a change within the cell, which ultimately makes the initial stimulus worse. This all started because we depleted ATP, and now we're further damaging the factories that make ATP. We're decreasing oxidative phosphorylation, which means we're decreasing the ability to make ATP from oxygen. The other problem is that as the mitochondria gets damaged, you get abnormal oxidative phosphorylation. I might have corrected this in your notes, but I don't have your version in front of me. If point 2, if it just says abnormal phosphorylation, you can add the word oxidative. I think that was missing in a version. I'm not sure if I fixed it in yours or not. So under membrane damage, the second point can say abnormal oxidative phosphorylation. What is the consequence of abnormal oxidative phosphorylation? Well, this sucker is going to be pumping out reactive oxygen species, which is in and of itself a vicious cycle because the mitochondria was damaged by reactive oxygen species. Now, the damaged mitochondria is causing abnormal oxidative phosphorylation, which is increasing reactive oxygen species, which is further damaging the mitochondria. Vicious cycles in pathophysiology is one of the primary reasons that things become irreversible, that we can't stop them because the stimulus is actually making the initial stimulus worse. So we're cruising through all these steps of cellular injury, and now we're starting to see that the membrane has been affected. We have defects in membrane permeability. Remember, the cellular membrane is a semi-permeable barrier between the intra and extracellular environment. As phospholipase has a longer and longer time to damage that membrane, it will eventually cause the membrane to become disrupted, right? It'll open it up. You will lose the barrier between the inside and the outside of the cell. At this point, cell contents leak out of the cell. Cell fragments are released. Lysosomal membranes have been disrupted. Digestive enzymes have been released, and they're just going to start chewing everything up around it. We are clearly far past any point where we could ever reverse this because the inside is basically exploded out. The last step is that we see damage to DNA and proteins. So the source of that damage, right, is the targeted attack from proteases and endonucleases, and then the non-discriminate damage caused from reactive oxygen species. We'll go into a little more detail on some of this in a minute, but at this point I'm going to leave that there in your notes. So we have now gone through all of the cellular changes of irreversible cell death, and these are the steps that lead to cellular necrosis. Cellular necrosis, the very beginning of the lecture on Wednesday, we called this death by accident. The cell was not planning on dying, but it was given an environment that it couldn't continue to live in. So this picture kind of summarizes, right, all the changes that we've seen so far. We've got the swollen cell, the loss of the structure, right? We see some blebbing coming out here because the cytoskeleton has been disrupted. The nucleus has been lysed. All that DNA damage has created a totally abnormal nucleus. The endoplasmic reticulum is swollen and the ribosomes have detached. We have kind of dysfunctional mitochondria, which is continuing to pump out reactive oxygen species and kind of not creating ATP, which is a huge problem. The time that it's been exposed to this stimulus has allowed for the disruption of membranes, like the lysosome is free to have all the digestive enzymes come out. We've actually lost the integrity of the wall here. Cellular contents are spilling out. We see cell fragments leaking out. We're going to circle back to this in a few minutes. But this expulsion, or this exposure maybe, of intracellular elements to the extracellular space is a really key element of necrosis. Whenever our body gets exposed to something that's not supposed to be there, for lack of a more technical way of saying it, the area where this cell is living is not equipped to have the intracellular environment outside of it. As soon as the intracellular elements of the cell spill out, the tissue around this cell is going to react to it. And it's actually going to react with inflammation. We are going to see the inflammatory response with cellular necrosis because the inside of the cell is being dumped out to the outside, and our body is going to react to that as almost like it's an invader and be like, whoa, 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 these guys are not supposed to be here. Let's call in our inflammatory response. So the physiological response to cellular necrosis is inflammation. And we're going to go through the steps of inflammation. We're going to talk about how our body recognizes these foreign invaders and calls the full inflammatory response. But this is important for how this looks when a patient is experiencing cellular necrosis. So we're going to take a minute to move outside of the individual cell and look at what would happen to the tissue, right? The collection of cells altogether of the body when cellular necrosis is happening. Let's look at the patterns of tissue, what it looks like when necrosis is happening. And there's some characteristic things that we see over and over and over again when cells are dying for a particular reason. You might possibly be asking yourself, why do we care about this? Why do we care what a tissue looks like when a cell is dying? What does it really matter? Well, in many, many cases in clinical medicine, we don't know what the problem is immediately. And we have to solve that problem with, subjective history, signs and symptoms, objective tests, and looking at some of these tissues to try to figure out what's going on. And because the whole tissue looks different given different kinds of cell death, it can provide a clue to the underlying cause of the problem. What's causing the cell death in the first place? So let's talk about these different patterns of tissue when they are undergoing cellular necrosis. And the first is coagulative necrosis. Coagulative necrosis is the pattern of tissue injury when the cell death is caused by reduced blood flow. Okay, so coagulative necrosis goes with reduced blood flow. This chunk here is the coagulative necrosis. The characteristic look is that it is very pale in color. The cells in this area are dead. Okay, so it's pale in color because the cells are dead. It's pale in color because there's no blood flow here. The other thing that's very obvious about coagulative necrosis is if you could put your hands on this and feel it, the tissue is very firm, all right? It hasn't lost its architecture. So the structure of the tissue around the cells is still intact, right? The architecture of the tissue is preserved. It's just the cells that are living within that architecture that are dead. So the tissue is not degraded yet. The cells of the tissue are dying. The shape of coagulative necrosis is characteristic in that it's typically wedge shaped. The reason it's this shape is that it follows the pattern of the blood supply network. So we normally have a larger vessel that branches out to feed the peripheral areas of an organ. So if this is where the reduced blood flow happened, it would be only in the downstream arterioles and capillary beds that stemmed off of that main branch. So it has this characteristic web shape. So coagulative necrosis is the tissue pattern you expect with reduced blood flow. It's basically how any one of our solid organs look when it has ischemia or an infarction, with the exception of the brain. And we'll talk about why the brain is different in a minute. But basically, any of our solid organs that get reduced blood flow end up in coagulative necrosis. The second tissue pattern is liquefactive necrosis. Liquefactive necrosis is caused from a bacteria or a fungal infection. Basically, there is like a liquidy center where the injury is happening. The reason it's liquidy is there is an acute inflammatory response happening at the site of tissue injury. So white blood cells are called to the scene because of the bacteria or fungal infection, and the white blood cells are going to create the edema and the inflammatory response and try to chew up the bacteria and end up killing the area of tissue around it as well. So a couple of terms to define. An abscess is an accumulation of pus in an enclosed area. So when liquefactive necrosis happens within a tissue, you can get an abscess because that's the liquidy center. That's the area where the acute inflammatory response is happening. It's the little pocket where the damage is happening. So we said it's an accumulation of pus in an enclosed space. I just want to make sure everyone knows what the word pus means. Pus is a viscous fluid substance. It's very kind of creamy and yellow, and it's formed at the site of inflammation because it's a collection of the fluid of the inflammatory response, the proteins that are getting broken down, and the dead white blood cells that are kind of sloughing off. So an acute inflammatory response causes fluid to accumulate, but we don't call that fluid pus until it starts to accumulate proteins and white blood cells. And then it becomes a much thicker, I hate to use the word creamy to describe something in our body that's gross, so I apologize. There's just no better word for it. I think we've all seen pus in our life, so you can picture the white creamy material I'm referring to. Okay, so liquefactive necrosis is what we see when the cell death is from bacteria or fungal infection. The exception here is the brain. The brain experiences liquefactive necrosis in response to ischemia. So remember, anywhere else in our body, if you have reduced blood flow, the tissue pattern you see is coagulative necrosis. In the brain, when you have reduced blood flow, we see liquefactive necrosis. It's very different. The theory is the reason that it happens, it might be due to the high amount of lipids in the area. And that when the lipids start to die, it actually breaks down and it kind of releases some of those intracellular enzymes that are breaking down kind of the tissue structure more than in other areas of the body. So we do see kind of one difference here in the brain. The next one to talk about is gangrenous necrosis. Now, it always bothers me a little bit to put this in your notes here because gangrenous necrosis is not a specific pattern of cell death, but it's a term that we commonly use in clinical practice. Okay, so it's not a specific pattern, but we still use it all the time and it has a descriptive purpose. It also is a term that we use to describe a limb that has undergone necrosis as opposed to a specific tissue bed. So coagulative and liquefactive necrosis, we're talking about what a tissue looks like. Gangrenous necrosis is a term that we clinically use to describe when a limb is experiencing necrosis, okay? So gangrenous necrosis is from a critically insufficient blood supply to a limb, and it can either be dry or wet. So dry gangrene is when you have reduced blood flow. So dry gangrene is actually coagulative necrosis. So it falls actually under the category of coagulative necrosis because it's cell death in a tissue caused from reduced blood flow. Where we see it clinically a lot, this is the type of gangrenous necrosis that occurs when someone has frostbite. You can see this in some cases in patients with diabetes who have very severe vascular disease and they're not getting blood flow to their feet. The symptoms of dry gangrene can be numbness and pain. The tissue turns from brown to kind of purplish blue to black. The tissue dries up and falls off. So it is a complete loss of the cells within this tissue. And for a while, the architecture is preserved, but it eventually just starts to dry and fall off. So that's dry gangrene, when a large section of your limb is not getting blood flow. Wet gangrene is when you have both reduced blood flow and a bacterial infection. And wet gangrene is the dangerous one. Like if you've heard people talk about, if it goes gangrene, it's a life-threatening thing. They're talking about wet gangrene when they're talking about that. Wet gangrene is the life-threatening problem because you have bacteria that have gotten into a limb that has poor blood supply. And the only way we can fight off a bacteria is through our inflammatory response. And our inflammatory response gets delivered to the area where the bacteria is through the blood. So if you have bacteria that's growing and having an infection, and you can't raise a defense against it because you have insufficient blood flow, the bacteria can grow out of control. And this is the case where a patient can experience sepsis and can lead to death because the infected fluid can actually re-enter their general circulation and go anywhere in the body. Okay, so this is a much more serious condition than dry gangrene because of the presence of the infection. So wet gangrene is very much a liquefactive necrosis because liquefactive necrosis is caused by bacteria or fungal infection. You get that, total degradation of the tissue. Wet gangrene is extremely painful. You can see swelling because of the inflammatory response. You can see blisters with pus, again, because of the inflammatory response. It typically has a very foul smelling discharge because of the death. The tissue typically goes from red to black, and then the medical team is always looking for systemic problems like fever and sepsis with gangrenous necrosis that is wet. The last tissue the next tissue pattern of when the cell is undergoing cell death is fat necrosis. Fat necrosis is a very specific tissue pattern that results when digestive enzymes act on fat. So we're really talking about cell injury to the pancreas because the pancreas, on the inside of the cells of the pancreas, they have powerful digestive enzymes because that's kind of the job of the pancreas. So in necrosis, the intracellular elements spill out. So when cells of the pancreas die and they spill their intracellular elements out, they release their powerful enzymes and it acts on fat. So the process here is actually what's called saponification. When fatty acids are exposed to calcium, they actually form soap. So fat necrosis is you can visibly see these white chalky deposits because of actual soap that is being kind of laid down and deposited as the cell death occurs. So it's a very specific pattern that occurs with pancreatic cell death. All right, the last one is caseous necrosis. And caseous necrosis is most typically caused from tuberculosis, but also in response to certain fungi. Caseous necrosis is kind of when the immune system is trying to protect you from an invader and it's having a hard time killing it. So instead of, if it's ineffective at killing the bacteria, the immune system actually builds like a little wall around it, and it creates a barrier so that the infection doesn't get to the rest of the tissue. So that barrier is typically made up of macrophages that surround the infection. Once that microbe is walled off, kind of wrapped around in this barrier of macrophages, you're protected from the infection. That sac, that little walled off infection, is called a granuloma. And the bacteria will stay in the body, but it won't spread because it's walled off within the granuloma. So caseous necrosis is really kind of a combination of coagulative and liquefactive necrosis because it has the infection, but in that area it also has the coagulative death. The tissue left behind ends up with kind of a cheese-like appearance because those like saccules kind of leave that open space. Ow, that, excuse me, just ripped my nail. Kind of leaves that kind of Swiss cheese appearance of caseous necrosis. So I made this chart to kind of give you a summary. Well, I shouldn't say I made it. It comes from your textbook. There is a summary chart of these different patterns of what happens within the tissue when the cell is dying, if this helps in your study and kind of keeping you organized. So we just went through tissue patterns when cells are undergoing necrosis. And these were the steps that we've walked through in the mechanism of cell injury. But in the very beginning, when we introduced the concept of irreversible cell injury, we said a cell can die from necrosis, death by accident, or apoptosis. And I want to add in this piece of how a cell could actually undergo apoptosis. Now, to help you remember the difference between these two, I'm going to give you a very gory example, because gory examples stick with me and then I remember them when I'm studying. Necrosis that we just went through, if you think of it, it's a type of death that's unexpected, right? So imagine, I'm standing in front of you in 356 and somebody walks in the door with a shotgun and blows my head off and bits of my brain go flying everywhere around the room. I would definitely die. And that is what necrosis is like. I had no idea it was happening. There is a stimulus that disrupts the integrity of my membrane, and my insides splurt out all over the place and create a huge mess, right? A disgusting, huge mess. That is cellular necrosis. It's a big, huge, disgusting mess of intracellular junk that goes everywhere, causes a big problem because the body has to respond with acute inflammation, and the cell's obliterated. Cell death by necrosis. Now, let's talk about how the cell could die from apoptosis. So apoptosis is planned death. So to keep my gory example going, apoptosis would be like if I looked around and was like, I'm done. I've had enough. And I build myself a coffin and I climb into it and I put my favorite outfit on and then I take 1,000,000 sleeping pills and I very carefully die within a box that holds me and I fall asleep and I am dead. That is apoptosis. I planned it. I organized it. I made it very clean and well packaged, but I still had the end result of death. Okay? Those are the two differences. I hope I didn't completely weird you out with my example and that you will remember it on the test. Okay, so let's talk about this very careful climbing into our coffin death. And it actually has a similar shared mechanism with necrosis, and that is the damage to DNA and proteins. So if the initial stimulus causes DNA damage, and things that can do this are drugs, chemicals, radiation, oxidative stress can all damage DNA. If the damage to the DNA is too severe and cannot be repaired, it can lead to apoptosis. And again, as it says in your notes, apoptosis is programmed cell death. The cell is going to activate enzymes within itself to degrade the cell's DNA and proteins. Within every cell in our body, we have a dedicated set of gene programs that are ready to activate apoptosis. There are coordinated, well-regulated, internally programmed series of events that can kill off a cell once a cell recognizes that it's damaged beyond fixing. And that usually happens when the DNA is so much damaged, because that's our blueprint. And the cell's like, no, my blueprint got so messed up. If you replicate me, it's going to cause some problems. So I am activating my death sequence and I'm taking myself out of here. So apoptosis happens a lot in physiology. So during embryogenesis, we have a lot of necessary apoptosis. We gave the example of all that hyperplasia in lactation. Well, then those cells undergo apoptosis when they're no longer needed. All of our immune cells, once they're done doing the immune response, they can undergo apoptosis. Like, I did my job, peace out, I'm done. The problem is that it also comes from a lot of pathological causes, and we went through a number of these already. So this is the cartoon version. I'm not going to draw all this out because the steps of this are a little less important than cellular necrosis because we don't interrupt it clinically at different places with treatments. but it's important to know that there are two separate pathways that are induced under very different conditions. So we have the intrinsic or mitochondrial pathway and the extrinsic pathway. So let's first talk about the mitochondrial pathway because it's listed first in your notes. That seems to work well. The intrinsic pathway will get activated if the mitochondria is kind of too leaky and the cell is not well. It can turn on the mitochondrial pathway. Basically, proteins inside of the mitochondria are released into the cytosol, and that triggers suicide programs in the cell. So in your notes, I listed a couple common causes of this intrinsic pathway, of the cell recognizing that it's not well and leaking mitochondrial proteins out. The other pathway, sometimes called the death receptor pathway, that's a very cool name, is when a signal is delivered to the apoptotic cell to die. So there's receptors on the outside of the cell that when they get bound are a signal to trigger the internal steps to kill the cell. This is how we get rid of like activated T lymphocytes, for example. They have a receptor. Once it gets activated, like you guys are done, your job is done, that triggers the cell for them to kill themselves. In either case, whether it's from mitochondrial proteins leaking out in the intrinsic pathway or an extrinsic signal, both pathways ultimately produce caspase cascade, or the execution phase. Through different series of steps, we all end up with the executioner caspase. I just love that this is named so exactly with what it does. I always love when that happens in physiology. I hate when it's named after the dude that found it, but when it's named the executioner caspase, oh, come on, that's so cool. Its other name is caspase 3, if you'd like to know. Way less exciting. Okay, executioner caspase is on the scene regardless of whether it's intrinsic or extrinsic. Now, this part gets important, okay? So bear with me. The executioner caspase will cleave proteins, okay? So we're going to start breaking down cytoskeleton. We've heard of that before, right? We're going to lose our cellular structure. It's going to degrade DNA, so further breaking down the DNA, the nuclear matrix, and then obviously that cytoskeleton kind of comes in with the proteins. So that's what it does. It cleaves proteins, it breaks down DNA and the nuclear matrix. What's missing here? A big one is missing if you compare this to necrosis. There is no attack on phospholipids. What does that mean to you? Do you already know the significance of that being missing in this step? If you do, you're on the steps to being able to put concepts together instead of memorizing a list. What is the significance that in apoptosis, there's no attack on phospholipids? Phospholipids are in the membrane. In apoptosis, nothing is attacking the membrane. The membrane is intact. So, the cytoskeleton detaches from the membrane. The elements within are being broken down, proteins and DNA are being broken down, and we can get blebbing, just like we did in necrosis, right? Where we get this outpouching because we've lost the cytoskeleton integrity. But now the blebs will actually get completely pinched off and create these apoptotic bodies. Little membrane-bound saccules filled with cellular components that can then be chewed up and degraded by phagocytes. In apoptosis, there's no leaking of the intracellular elements. They're packaged up in little apoptotic bodies and destroyed. So to give you that cartoon version, like we did in necrosis, from the normal cell, the apoptotic cell looks very different. Number one, the cell shrinks. right? In necrosis, it gets bigger because it gets engorged with water. Here it shrinks because it's packaging up its cellular products and goes away. So the cell gets smaller and smaller and smaller and smaller. We see fragmented DNA and condensed nuclear chromatin as that DNA is fragmented. Can I say DNA frag on mine? Does that make sense? We see the blebbing that occurs because of the loss of cytoskeleton, but instead of the membrane being attacked, the membrane stays normal, so it's able to actually pinch this completely off and create a fully intact apoptotic body surrounded by cell membrane that can be phagocytized and destroyed. These apoptotic bodies have what's kind of like called like an eat me signal on their membrane. So it triggers just tissue macrophages to phagocytose them. There is no inflammatory response here because as far as the extracellular environment is concerned, nothing is abnormal here. There's no invader, right? Because everything's still packaged within the membrane. In necrosis, the membrane got disrupted, so all of this stuff that lives inside of a cell is suddenly outside of it, and the tissue's like, whoa, bring in the inflammation. This is a huge mess. In apoptosis, since it's still wrapped in membrane, nothing sees that it's happening, and they just get phagocytized and taken away. Oh, this is kind of a cool picture. So this is a picture of a cell going through apoptosis, and you can see all the apoptotic bodies just floating away to be phagocytized and destroyed. So this is just a picture that kind of goes through the phagocytosis of the apoptotic body. The macrophage literally just engulfs the apoptotic body. And then once it's within its membrane, it can degrade it. It's pretty cool stuff. All right, so we've gotten a summary of necrosis versus apoptosis so that you have all of the elements side by side. Hopefully this will help with your studying and to make sure that you can tell the difference between the two. When you are studying any step that, especially if I added it to a drawing, or made a little emphasis about it, can you answer the why? Why is this step important? Why does this step ultimately help the goal of killing the cell or saving the cell, depending on what you're talking about? Challenge yourself to think about the why. Could go through this section and study and then go back to this chart and go through each thing. Cell size, necrosis. Can you exactly explain why the cell gets enlarged in necrosis and shrunk in apoptosis? Can you tell me the exact steps that disrupt the membrane in necrosis but leave it intact in apoptosis? Can you talk about, the why there is or is not inflammation? Can you go through each one of these and say the why and explain the difference between the two? If you can do that, you are ready. Like, this is the level of understanding necessary. If you have to memorize the steps of necrosis, 1, 2, 3, 4, 5, 6, 7, and I have to list them in order, and if I start in the beginning, I can get all the way through them, then you're not at a place where you're thinking about this and considering the steps. And I challenge you to go to each one of those steps and ask yourself the why, or have a friend quiz you on the why and see if you can say it. Okay, for our first week, I'm at a really good place to break, and I'd really love to do fluid dynamics in person all at one time. So we're maybe 8 or 9 minutes shy of our full time. But I hope that you take that time to, clean up your notes, look up any words that if I said a word and you just didn't know what it was, at any point in these videos, take the advantage of remote learning while we got it. You can pause the video and quick do a Google search. And right now, go back into your notes. If there was any place that you're like, whoa, I wrote a lot down there, just reread it. What you wrote, does it make sense? You will be in a better place when you come back and study your notes. All right, I have thoroughly enjoyed this time. I can't wait to see you guys on Wednesday. Any of you who are listening to this on Friday morning, don't forget, I will be online at noon on Friday. The Teams link is in your D2L page under content. I'll also include it in the email when I send you these links. which you've already gotten. That's happening in the future for me. It's happening in the past for you. This is a real mind bender, this whole remote learning. All right. Have a wonderful day, you guys. This has been great. See you on Wednesday.